My name's Gemma Hartley. I'm a PhD student in the Department of Immunology at Monash University, and I'm here to talk about our recent publication in Allergy called "The Second COVID-19 mRNA Vaccine Dose Enhances the Capacity of Spike-Specific Memory B Cells to Bind Omicron BA.2." To examine the response generated by primary Pfizer vaccination, we recruited 30 healthy individuals and took whole blood samples pre-vaccination, 3 weeks after the first dose, and 4 weeks after the second dose. In the lab, we generated spike receptor binding domain protein to probe the antigen-specific response. By ELISA, we can see all individuals generate high levels of RBD-specific antibodies after the second dose, and using a pseudo-neutralization assay, all individuals show neutralizing levels of antibodies after the second dose. Receptor binding domain protein was also used to create RBD tetramers to identify antigen-specific cells. Double discrimination of these tetramers allowed us to identify true tetramer-positive cells, and this, alongside an extensive immunophenotyping panel, allowed us to identify subpopulations of class-switched memory B cells. After the second dose, we see a large expansion in the absolute number of RBD-specific memory B cells, and this is largely due to the expansion in IgG1 class-switched memory B cells. Finally, we wanted to examine the capacity of primary Pfizer vaccination to recognise variants of concern. In this study, we examined the response to Beta, Gamma and Delta, and also the highly mutated Omicrons BA.2 and BA.5. By ELISA, we can see the RBD Wuhan-specific antibodies significantly increase in their capacity to bind Beta, Gamma, and also Omicron BA.2 and BA.5. Alongside this, we also generated RBD variant tetramers of Gamma, Delta, BA.2 and BA.5, and we can see that although the proportion of cells that recognise Omicron's BA.2 and BA.5 are low, we can see the second dose increases the proportion of cells able to recognise BA.2. To summarise, the second dose of Pfizer vaccination generates high levels of RBD-specific antibodies and neutralising antibodies, a robust RBD-specific memory B cell response that is predominantly IgG1 positive. The second dose also increases the reactivity of antibodies to Beta, Gamma, and Omicron BA.2 and BA.5, and also the reactivity of memory B cells to Omicron BA.2. Therefore, this suggests that the third and fourth dose boosters provide protection by increasing reactivity to variants of concern. Thank you very much for listening today.